Thanks for joining, everybody. Good morning, and uh, thanks for joining in the morning for the today's session. I would like to begin by acknowledging the Wurundjeri people of the Kulin Nations as the traditional owners of the land on which our Parkville campus stands. I also acknowledge the traditional custodians of the various lands on which you are all joining me from today. I pay my respects to the elders past, present, and emerging, and to the Aboriginal elders of other communities. I would like to begin by introducing myself. I am Inamur Rahman, a CCMP PhD student of the University of Melbourne Node. I am working under the supervision of Dr. Isabel Ruila. Before starting the today's lecture, I would like to remind few housekeeping rules. I would like to ask audience joining over Zoom to stay muted throughout the presentation and to leave all questions to the end of the seminar. I would also like to ask audience to type it in the Zoom chat during the presentation if they want to be read out later. It is to inform you that the seminar will be recorded and the recording will be shared on CCMP website. So I would like to welcome Dr. Steve Rico. Uh, Dr. Steve Rico is an associate professor at the Department of Chemical Physiology and Biochemistry, Vollum Institute, Oregon Health and Science University, Portland, Oregon, USA. He completed his PhD from the University of Washington in 2006, followed by a postdoctoral stint at the Howard Hughes Medical Institute. There he embraced and learned cryo-EM to investigate membrane channels and dynamic protein assemblies. In 2014, he began an assistant professor at Portland State University and now working as an associate professor. In 22, Rico joined Oregon Health and Science University, holding dual roles in the Department of Chemical Physiology and Biochemistry at, and the Vollum Institute. His lab combines biochemical techniques with atomic level cryo imaging to explore protein function and disease. Rico has been involved with the development of the Pacific Northwest Cryo-EM Center and now serves as the OHSU lead investigator for the PNCC. As you all know, the topic of the today's talk by Rico is mechanistic insight into gap junction cell-to-cell communication enabled by cryo-EM. So over to you, Dr. Steve.
Great. Thank you so much for that nice introduction. I want to thank Aidan Grosses for the nice extension of the invitation to speak here today, and Tracy and all the organizers. It's a real pleasure to get to share our research with this really wonderful community that you all have built. So the plan for my talk today is really kind of to give both a historical perspective and then move on to a more modern view of how cryo-EM has really served as an enabling technology for really many decades toward understanding the structure and mechanism of one of my favorite biological systems, and that's the gap junction communication channels. And I hope this story sort of serves as an illustration of how the field of cryo-EM has grown over the years, and how it's contributing to the field of molecular biology, and ultimately leave you with a sense of how much more room there is for the field to grow and to contribute for many more years to come. And so the system that we study, the gap junctions, really has a unique place in the history of electron microscopy, and it's because the system was actually discovered through the use of an electron microscope. And it was discovered through images much like this one shown here. So some of you may be familiar with the role of gap junctions in forming what's known as an electrical synapse in the brain. And that's actually what you're looking at here. This is an early electron micrograph taken by Camilla Paracchia back in 1973. This isn't the first image of a gap junction, but to me, it's one of the most striking. And so for those of you who may not be familiar, what you're looking at here, these horizontal striations are the plasma membranes of two communicating cells. And these punctate densities are individual gap junction channels that effectively stitch these two communicating cells together. And in doing so, they form this characteristic 3.5 nanometer gap. And it's this morphological feature that was first identified on electron micrographs that led to the discovery of these channels. And so I refer to these channels as communication channels because that's the function that they play in the roles of these cells. And they're able to serve as effective communication devices based on their unique molecular architecture. And so I want to explain, I think, what are 3 key features that lead to these channels to be so effective in performing this role. So the first is that they're integral membrane proteins, and they pass not one, but 2 membranes. So they pass through the membranes of both of these communicating cells. And in doing so, they establish a continuous pathway for the cytoplasms of both of these cells to exchange chemical information. And so these channels establish the second kind of unique aspect of their structure is they establish unusually large pores. So the pore of these channels, at least for a molecular scale, is unusually large. So it's about 1.5 nanometers in diameter. And so that's sufficient to pass not only electrical information between cells, but also chemical information. So things like small molecules, nutrients, hormones can readily pass through these communication pathways. And the last thing that I'll point out is you'll notice there's a large number of these channels. They form these large arrays, or in our field, we call them gap junctional plaques. And so these plaques can be formed by hundreds, if not thousands, of individual channels that self-assemble into these large assemblies. And in doing so, they form very efficient communication pathways that allow for the near instantaneous passage of electrical and chemical information between these 2 communicating cells. And they not only facilitate communication between neighboring cells, but effectively establish a syncytium that couples together cells in an entire tissue or organ. And I think that's illustrated well with these live cell imaging that I'm showing here. So the gap junctions not only form electrical synapses in the brain, but they also serve this function in the heart. And these are the proteins that are responsible for the synchronous contraction of the muscle cells inside of your heart. And along with electrical synapse formation and neurons, they also play important roles in glial cells, for example, by facilitating long-range calcium signaling in these cells. And that's what's illustrated here on the right, these long-range calcium transients. And so the gap junctions not only play fundamental roles in electrically active cells, but they're expressed throughout our body, where they play fundamental roles in development and physiology in almost every cell in your body. And in my lab, we use the eye lens as a model system for studying and understanding the role of gap junction biology. And so I'm not going to get really deep into lens biology today, but I want to point out a couple of unique and what I find fascinating features about this organ. And so the lens is unique, right? It has a unique function inside the body, and that's to focus transmitted light for vision. And to do that effectively, it has to be completely transparent. And so to enable this transparency, it's evolved several unique cellular adaptations. And one of them I'll point out is that your lens actually lacks a traditional vascular system, right? And of course, a vascular system would interfere with transmitted light. And so instead, it's evolved this unique, what we call a microcirculatory system that's established entirely by membrane channels and transporters that effectively deliver nutrients deep within the lens tissue and then circulate waste products back outward through these gap junction channels, where these play a central role in this circulation process. The other aspect that I'll point out is because of how the lens is developed, the cells inside of your lens actually never turn over. And in fact, the proteins inside your lens cells never turn over either. And so what that means is that the protein that was synthesized in your lens from the day you were born has to stay with you throughout your entire lifetime and remain functional in order to maintain transparency of this organ. Of course, this is a very hard thing to do for many decades. For any of the biochemists in the audience, you'll appreciate how hard you might have to work to keep your protein happy for just a couple of hours to perform whatever experiment you might be trying to do. But in the lens, these proteins remain functional for many, many decades. But eventually they succumb to a lifetime of environmental stress and chemical damage that ultimately leads to the major disease of the eye lens, which is cataract formation, which, despite effective surgery, still remains the leading cause of vision loss and blindness in the world. And so my lab is very interested in trying to understand the molecular basis of these aging effects that happen within the lens and how they affect protein structure and function and lead to this disease. And because of the central role that the gap junctions play in many different physiological processes, their misregulation or aberrant function is associated with a wide range of different types of human diseases that range from blindness to deafness, arrhythmia, stroke, and some cancers. And because of these important roles in health and disease, they've been a long-sought target in the field of structural biology. And in particular, they've been targeted by electron microscopists for a particular reason, and it has to do going back to their propensity to form these large two-dimensional arrays. And so these are actually some of the longest studied membrane channels in the field of structural biology, and in particular by a niche of electron microscopy known as two-dimensional electron crystallography. And so shown here on the left are some of the earliest images of two-dimensional crystals of the gap junctions. These little white donut-like structures are the individual channels. So these are studies done early on by Don Casper and colleagues. And as this technology developed over the years, so did the amount of structural information that we're able to coax out of this system. And so by the mid-80s, Nigel Unwin had improved the diffraction data on these systems and we could glean more structural insight. And so at the center here, you can now resolve the pore of these channels. As I mentioned earlier, they measure about 1.5 nanometers in diameter. So this is sufficient to pass the types of chemical information that they're involved with in the cell. And in addition to that, you can start to resolve the subunit architecture. And so you can count 6 different subunits in what we call a hemichannel, which forms in each of the plasma membranes. And so if we move over here to this 3D reconstruction, we have one hemichannel in the membrane here, another hemichannel in this membrane, and they come together and dock through these extracellular domains. And in doing so, they establish a continuous pathway. And so this reconstruction in the late 90s was done by Vincent Unger and was resolved to a resolution of around 6 to 7 angstroms. And so at this resolution, we can start to resolve secondary structural features. These sausage-like densities are the transmembrane alpha helices. And then I know that, with regards to current cryo-EM methodologies, this type of resolution may not seem too impressive, but at the time, this was really a landmark achievement in the field because there had really been very limited understanding of membrane protein structures at this time. Now, modern applications of electron crystallography can resolve biological specimens down to the angstrom level. And so this technology is not really limited in resolution. It doesn't really have a resolution limit that's relative to biological systems, but for the gap junctions, this methodology was really never able to go beyond this point in resolution. It's sort of 6 to 7 angstrom regime. And ultimately, if we want to understand the details of molecular mechanism, we want to be able to build atomic models. And to do that, we need to have resolutions that are much better than this. So something better than around 4 angstrom resolution, where we can resolve amino acid side chains and begin to build atomic models. And as it turns out, the next big leap in this field didn't come from electron microscopy, but rather from X-ray crystallography. And so I'm showing here the first X-ray structure of these channels. What I think I forgot to mention is at the subunit level, we refer to these proteins as connexins. And so there's many different connexin isoforms in humans. There's, I think, 21 isoforms in humans, and they're labeled by their molecular weight. So this is connexin 26 because it's a 26 kilodalton molecular weight protein. And connexin 26 has really been kind of the model system in the field. And so what you can see from the structure, so it was resolved at 3.5 angstroms, is sufficient to build a suitable atomic model. We have the 6 subunits in each of these hemichannels. This defined what the pore lining alpha helices were and gave us insights into these extracellular loops that form the docking domains and have specific interactions that facilitate this junction formation. We rotate the channel, so now we're looking down the pore, and you can see it's an unobstructed pore pathway measured at around 1.4 nanometers in diameter. And I'm going to point out an additional domain on here that will be a bit of a focus for my talk today. And that's an additional alpha helical domain that we refer to as the N-terminal helix domain, or NTH domain, that in this conformational state, it folds back into the pore, contributing to the pore vestibule. And this alpha helical domain is connected to the rest of the transmembrane domain through a short loop. And this motif is thought to function as a gating module for these channels. And the details of this gating mechanism are still trying to be resolved, but the idea is that in response to physiological signals such as voltage, that you would cause a lifting of this alpha helix and obstruct the pathway of the channel and result in a closed conformation. And so this structure really served as an archetype for the field for nearly a decade. And really no further new structural information was able to come out of this field for many years. And this really reflects not just a limitation in the field of gap junction biology, but it was really an overarching limitation in the field of membrane protein structural biology. And that's because of the many inherent limitations that crystallographers face in trying to work with membrane proteins. And so there's many obstacles in this field. These proteins were often difficult to express to the and purify to the high levels that were needed to grow 3D crystals. Detergents that were used to isolate these proteins from the native membrane environment were problematic to crystallographic studies. And if you were studying native proteins, the inherent heterogeneity that was associated with these native systems were also very problematic for crystallographic studies. And so this was really kind of the state of the art up until around 2013, when what we know now as the cryo-EM resolution revolution that took over the field of structural biology. And so this image that I'm showing here has really become sort of the hallmark, symbolizing this revolution in the field of single particle cryo-EM. But I want to remind us all that this is really a culmination of many different factors that kind of coalesced around the same time. So this had to do with improvements in microscope hardware, the new types of detectors that were coming out, as well as improvements in software algorithms to really determine the structures of these proteins with high accuracy. But I think the first demonstration of all of these technologies and the impacts that it was going to have on the field of structural biology really did come from Yvonne Chang's lab when they resolved the structure of TRPV1 at around 3.5 angstrom resolution, suitable to build an atomic model from a small membrane protein that had never been determined by any other methodology before. And so it was immediate to the field that cryo-EM was going to have a tremendous impact, not only on the field of structural biology, but a particular impact on the field of membrane protein structural biology, and contribute to understanding the physiological and roles in disease that these proteins play. And so it was also around this time when I was just starting my independent laboratory, and my hope was to be able to return back to cryo-EM and use this as a technology to gain new insights and really push the field forward in understanding gap junction biology. And so that's what we set off to do. And so this project in my lab really started off focusing on where all structural biology projects, at least using single particle cryo-EM, need to focus, and that's on the biochemistry. And because we're interested in the physiological changes that happen to these proteins in the lens, we really wanted to isolate these channels from native source. And so that's the protein that we work with. We have shipments of hundreds of eyes sent to our lab, and we dissect out the lenses and isolate the proteins from these native organs. And through a lot of hard work in traditional biochemistry, my first two trainees, Susie and Jeanette, were able to come up with a very nice purification strategy to isolate these channels. Now, another aspect that I haven't mentioned yet is that in most tissues, the cells express more than one isoform of connexin. And so in the lens, there's two isoforms that are expressed, connexin 46 and 50, which I've labeled here. And in fact, many of these isoforms have the ability to co-assemble in complex ways to form what we refer to as either heteromeric or heterotypic type gap junction channels. And we showed by cross-linking mass spectrometry studies that, in fact, these two channels do co-assemble into the same channel. So we have this heterogeneous mixture of these two isoforms. But, you know, coming from one of the great things about cryo-EM is once you've worked out your purification, you're ready to start studying it. And all you really have to do is put it onto the electron microscope and start taking pictures and let the data tell you how good your protein is going to behave and if it's going to be suitable for structure determination. And we were lucky to find out that this preparation was going to lead us to a very nice initial structure. And so this is the first 3D reconstruction we're able to get from these channels. We resolved it to just under 3.5 angstrom resolution. I think this group is relatively familiar with cryo-EM, but if there's any new people here, these you know, small protuberances or these features that you see are the amino acid side chains that are poking out on this 3D map. So we knew that it was going to be suitable to build an atomic model. Before I get to that level of detail, I'll just kind of walk you through some of the main features of this map. So we resolve an unobstructed pore of around 1.5 nanometers, and we clearly resolve this N-terminal helical gating domain in what is ostensibly an open state conformation. And then so to interpret this data, we can isolate out just a single subunit, and using tools that have been developed in the field of X-ray crystallography, we can interpret this data by building in our atomic models that best fit to the electron density map. And so at this point, I'll remind you that we had this heterogeneous mixture of 2 different isoforms. And as it turns out, Jeanette was able to build models for both connexin 46 and 50 into this same cryo-EM map. And the only reason she was able to do that has to do with the very close sequence similarity between these 2 isoforms. And so in light blue, I'm showing connexin 46, and in dark blue, it's connexin 50. In fact, these 2 isoforms are 85% identical, and obviously share a very close amount of structural homology as well. And so we were able to build models representing both of these isoforms using the same map. Okay, so at this point now, we had our structure. And so the question is, now what? And as I teach my students that the structure really shouldn't be the goal. The structure should really be the starting point. And at this point, I think it's also important to remind us all that of this, what I think is a very suitable quote from George Box, and that's that all models are wrong, but some are useful. And so we really wanted to set out to understand how useful this model is going to be in explaining some of the functional features of these channels. And so one of the aspects that has always kind of fascinated me with these channels is, despite establishing this very large pore and the capability of permeating many different types of substrates, they still demonstrate a relatively remarkable amount of selectivity. However, understanding the selectivity mechanism was going to be unique from kind of the traditional types of channels that were, you know, sort of the textbook examples of selective membrane channels, like the potassium channel or aquaporin water channel shown here. And so these channels are highly selective for just a single substrate, and they form a permeation pathway that is exquisitely matched at the chemical level to that particular substrate. And because of this specificity for substrates in strictly defined permeation pathway, these substrates can actually be resolved in experimental density maps. And that wasn't going to be the case for the gap junctions. There's many different pathways that substrates could take through here. And so in order to define the permeation pathway, we knew we were going to have to turn to a different type of methodology. And to gain insight into that, we decided to turn to what I like to refer to as another type of microscopy, or computational microscopy, but more quickly known as molecular dynamic simulation. And so here's an example of one of these simulations that we've run on these channels. And so we start the simulations by surrounding the channel on both sides with a mixture of ions. In this case, we had potassium chloride. And we let the simulation run, and what we're very pleased to see is, and from what you can tell in the simulation, is that the channel fills with these blue ions. These are the potassium ions, the positively charged ions. And every once in a while, you'll see a chloride slip into the channel. And the overall ratio, if you quantify this, is a selectivity of around 20:1, which is very similar to the selectivity ratio that have been shown for these channels by electrophysiology experiments. And so these MD simulations are extremely information rich, and I could really give an entire talk about all the different things that we observe in these simulations. But I'll just point out a few interesting things and to explain how we kind of interpret some of this data. And so one of the things you'll notice is that these ions, once they're in the pore, they don't spend the same amount of time in different regions of the channel. And in fact, in some regions of the channel, they seem to bind to these transient binding sites and spend a significant amount of time. Well, in other regions of the channel, like up here, the ions really spend very little time. And so you'll see if an ion gets into this region of the channel, it traverses very quickly to the other side. And so one of the things that we can do is basically count these events and use Boltzmann's statistical analysis to develop a free energy diagram that describes the permeation pathway of these channels and really move towards a more quantitative way to compare and contrast the behavior of these ions in these two different isoforms. And so one of the things that we showed is that not only is the relative selectivity between cations and anions relevant to what's been observed experimentally, but the differences in energy barriers between connexin 50 and 46 also seem to match experimental data very nicely. And so what you, just to explain this a little bit more, is these troughs here are the low energy sites. So these are these transient binding sites, and these higher energy barriers are the places where the ions don't like to spend much time. And you'll find that the highest energy barrier really is up towards the entrance of this channel, right up here. And structurally, this aligns to this N-terminal helical domain that I've been talking about earlier. And so we led to this kind of this idea that ion conductance and specificity is really controlled by this, quote, electrosteric feature that's defined by the N-terminal helical domain. And so I'll get into a little bit more detailed analysis of this domain in this slide here. And so a couple of features that I'll point out is in the conformation that we capture by cryo-EM, this domain forms kind of a classical amphipathic alpha helical structure. And so there's a hydrophobic face and then a hydrophilic face. And the hydrophilic amino acids are pointed towards the pore vestibule. This is fitting with the idea that these residues have been implicated in substrate selectivity and voltage gating. And in fact, these are the residues that are most variable between different isoforms, fitting with the idea that different isoforms have different degrees of selectivity for different types of substrates. And then these hydrophobic amino acids pack against the transmembrane domains, really kind of holding the conformation of this domain as we in this open state conformation. And if you look at the conservation of these amino acids, they're highly conserved throughout different types of isoforms, really kind of fitting with this conserved structural role. And so this all seemed to make sense, but in comparison to the previously described X-ray crystallographic structure, in fact, this is the part of the channel that was most deviated from the connexin 26 structure. And in their model, you can see that they had many of these hydrophobic amino acids displayed in completely different orientations from what we resolved in our structures. And some of them were even pointing towards the permeation pathway of the channel. And, you know, ever since I started studying this structure, this part of the domain always kind of caused a question in my mind, and it brought me back to the first structural biology course I ever took as a graduate student, taught by Wim Hol, and he taught me that, you know, greasy groups like greasy grooves, and so having these greasy groups nowhere near a greasy groove really suggests that this wasn't a very energetically favorable conformational state, and this becomes really evident when we compare the molecular dynamics simulations from all three isoforms. And so we simulated the entire channel. I'm just highlighting the N-terminal helical domains. And you can see that effectively, as soon as we start the MD simulations on Connexin 26, this domain completely falls apart and unfolds. And so while we still don't fully understand what physiological context the conformation of Connexin 26 may be representing, what we've kind of concluded is that using cryo-EM, we were able to capture 4650 in this what we call stabilized open state conformation. And we've continued to interrogate the role of these hydrophobic anchoring residues through functional mutation studies together with Donglen Bai. And so we're pretty happy with how that was explaining the role of those residues in stabilizing the open state of these channels. And so the other thing that we really want to understand is if our model could help explain some of the phenotypes that were observed with disease-causing mutations of these channels. And so it turns out that the gap junctions are the most commonly observed mutation of proteins associated with what's known as congenital cataract. So this is an inherited form of the disease that has an early onset in life. And if you map these mutation sites to the structure of our protein, you can identify certain hotspots of most common mutations. And so some of the effects of these mutations are readily explainable. So for example, mutating these extracellular loops can lead to the loss of junction formation and the uncoupling of these channels. Mutations inside the pore often fall at some of the very same residues that we saw forming transient interactions with the ions in the permeation pathway and are fitting with the observed changes in the conductance of the channels with mutations at these sites. However, some of these mutations were, their functional effects were a little bit more puzzling, and I'll give you an example here. So this position T39 in Connexin 50 can be mutated to an arginine that's associated with cataract formation. And our collaborator brought this particular residue, this particular mutation to our attention because the phenotype was a little bit hard to explain. And so what our collaborator, Lisa Eberhard, had measured is that when she introduced this mutation, she saw a complete loss of voltage gating response in these channels. And so because we isolate from native source, we're not really able to introduce these mutations for structural analysis very easily. And so we again turned to molecular dynamics simulation and we simulated the effect of this mutation. And what we saw was actually very satisfying. And so this amino acid on the N-terminal helix, this aspartate at position 3, is normally pointed upward in the wild type channel. But when we introduce an R39 at this mutation site, D3 flips downward, which is negatively charged and forms a salt bridge with the positively charged arginine 39. And what we believe is happening here is that arginine 39 is effectively neutralizing this voltage sensor and locking it in the open state conformation. And this is leading to this loss of voltage sensitivity. And furthermore, this interaction occurs between two neighboring subunits. And this really seemed to help rationalize the dominant phenotype of this mutation. And so at this point, we felt very comfortable with our structure and felt that it was offering useful information and interpreting some of the functional effects of these channels. And we're ready to move on to do some further interrogation by structural biology. But at this point, before we kind of moved on to much more complex things, I was really kind of dissatisfied with a certain aspect of our biochemistry. And that has to do with the fact that I mentioned that for membrane channels, we have to isolate them from their native lipid environment. And we typically do this with detergents. And our first EM structure was done with a detergent analog, an ampopole. And we knew from prior studies that the local lipid environment can impact the architecture of these channels and augment the function of these channels. And so we really wanted to be able to put these channels into a more native context of a lipid bilayer. And so at this point, we decided to turn to using what were at the time these kind of emerging use of lipid nanodust technologies. And again, this audience some of you in this audience may be familiar with these, but just in case there's people new to the field, there's lots of different flavors of these lipid nanodisk technologies. The one that we've had the most luck with are known as membrane scaffold proteins. These are really just engineered variants of apolipoprotein. So they're amphipathic alpha helices that, when mixed under the right conditions in the presence of lipids, can self-assemble to form these puck-like structures that effectively solubilize a patch of a lipid bilayer. And by having them in the soluble form, they can tumble in solution. So they became very attractive to electron microscopists, because if you perform this reconstitution experiment in the presence of your favorite membrane protein, you can coax your membrane protein back into this lipid bilayer environment and still maintain all the rotational degrees of freedom that you need to record the images and reconstruct a three-dimensional structure. And so at the time, I had this very talented undergraduate student, Kimberly Dolan, who put a lot of diligent effort. I think she spent almost a year screening various types of MSP scaffolds and tuning the lipid to protein ratios of these conditions to get the biochemistry down just right. And once she had that biochemistry worked out, she worked closely together with her mentor in the lab, Jonathan Flores, and together they went to image these reconstituted channels by electron microscopy. And so here's an example of what this data looked like. So on the left, I'm showing you a micrograph, and on the right, these 2D class averages. And of course, the biggest limitation in single particle cryo-EM is this low signal to noise. And so we overcome that through brute force and collecting hundreds of thousands, if not millions of individual particle images. And we average them together to get these two-dimensional projection averages shown here. And once we started seeing these projections averages coming out, we really started to get very excited. In fact, this is still my favorite part of the image processing routine. This is when I'm hanging over my graduate student's shoulder and watching this data come in. And when this particular data set came in, we started to get very excited. And so I'll zoom in on this particular 2D class average to explain some of the features. So we could readily identify the two nanodiscs. So we had successfully put the channel into not one, but two nanodisc structures. And if you measure the separation distance between these two lipid domains, we measure 3.5 nanometers, consistent with what we'd see, what we would have expected. But in addition to that was all these high resolution details that were starting to be resolved even in 2D classes. And so you can see this beautiful lattice-like structure that is forming these docking sites, these vertical densities of the transmembrane alpha helices. But in addition to that, you can see these horizontal striations shown here and here. So these are actually representing the helical pitch that was being resolved in these transmembrane alpha helices just from the 2D classes alone. And so we really knew from these early processing that this structure was going to do something special for us. And I was so excited that I had to send a text of these images to my wife, Susie. And so Susie actually works in my laboratory and she was part of this project. So she knows what she's looking at. And I think her response is just perfect for what happened next. So she says, "Holy shit, what an angstrom." And if you know my wife at all, you'll know that she really never swears. And so this is really out of character for her. Me, on the other hand, I have a little bit looser relationship with the English language. And so I'll let you read my response. But I think ultimately this really served to highlight just how excited we were about this data, because ultimately through a lot of hard work and effort on the image processing pipeline, Johnny was able to ultimately resolve these channels down to 1.9 angstrom resolution. And so I'm showing here a zoomed window, and here you can see the beautiful detail that was resolved in this density map, clearly resolved rings through the aromatics. And so he was able to build a highly refined atomic model into this density map. But for me, what was really exciting is all the additional features that we're able to resolve at such high resolution. And so these red punctate densities are all the ordered solvent molecules that are bound to the channel. And of course, given the motivation for this work, we're very excited to see these blue rod-like densities, which are the ordered lipids bound to these channels. And then ultimately, we're able to build this atomic model of this highly refined atomic model of the gap junctions, the lens gap junctions. And so I know that this group, at least many of you in this group, will have some more experience in electron microscopy. And so I want to take a little bit of an aside and just point out a couple of lessons that we've learned from this project and from collecting many other data sets on this protein since, that I think are informative and hopefully helpful to those of you who are maybe embarking on your own cryo-EM projects on your favorite membrane protein. And you know, this we've been able to collect data sets on this system on many different types of hardware, different types of camera, energy filters, without energy filters, etc. And while some of these things can make marginal improvements, I really just want to emphasize the importance of grid optimization, because in our experience, this really has the biggest impact on the outcome of the final reconstruction. So I'm showing you here an electron micrograph that we've collected on a very similar prep to what I've showed you before. But if you do a reconstruction on this data set, it'll only go to about 2.6 angstrom resolution. And that's because the ice thickness on this particular sample isn't as optimized as it could be. And so if we had worked harder to optimize the ice a little bit better, like in the micrograph shown here, this data set will easily go down to 2 angstroms and below. And I hope you can appreciate the difference in contrast that you see in these particles, the greatly improved signal to noise that you can get by really focusing on optimizing the ice thickness of your cryogrids. The other aspect that we kind of learned in processing this data the first time around is so crunchy particles. Sorry, this has become sort of a nickname for when we get grids that have these particles that seem to just kind of pop off of the micrograph. So we're always looking for crunchy particles. But the other aspect that we learned is the importance of low defocus. And so when Johnny was processing this first data set, we got to our final round of 3D classification, and we saw our particles were falling into these 2 different classes. And at the structural level, these 2 classes are identical. We couldn't tell any difference between these 2 classes. But one of them that had the most amount of particles could only refine to would refine to 2.2 angstroms, but the other much smaller class refined to 2 angstroms, and then after our final polishing is what led to the final map at 1.9. And so there's only 26,000 particles that went into that final map of the entire data set. And for a while, we really struggled to understand what was separating these 2 classes. And it wasn't until I think we got a tip on Twitter to go look at the defocus values of these 2 classes. And in fact, and I wish I could remember who gave me this tip because they were right. When we went back and analyzed the defocus values, the smaller class was the class with all the lowest defocus values. And so that really highlighted to us the importance of really using low defocus. And so now when we set up our data collections, we really try to keep the defocus as low as possible. So between like minus 0.5 to minus not much above 1 micron defocus when we collect data sets now. All right. Happy to discuss any questions about this at the end. So going back to the structure and some things that we're learning at this higher level of resolution. First thing we want to do is check the N-terminal helical domain. It was in the same conformation we'd seen previously, so we're happy to see that. In fact, most of the protein structure was in the same conformational state that we'd seen in our amphipool system. And so we turn up to the other features. We resolved over 400 water molecules. You know, for me, this was really exciting. I come from being introduced to membrane biology by studying water channels. I have I have a close affection for water molecules, and I think all biochemists should have a close affection for water molecules because they play an integral role in the structure and function of all protein structures. In fact, when I teach biochemistry, I teach my students that water is the 21st amino acid because of its fundamental role in protein structure. But rather than getting into the details of what we think some of the waters are doing, I really want to turn attention to the lipid molecules, because as a membrane protein biochemist, the only thing I like more than water is lipids. And so, oh, right, sorry. So I think the other important thing about water is, just to make one more note, is from the point of view of structure-based drug design. And so understanding where these water molecules are is very important for drug design, because if you can design drugs that displace these waters, you can often get a large increase in the delta G of binding by releasing those waters back to the system through entropic gains, right? So important for structure-based drug design and being able to reach these levels of resolution. Okay, so back to the lipids. And so I'll remind you that these nanodiscs generate a lipid bilayer, but the only lipids that we're resolving could be mapped to just a single leaflet of the lipid bilayer. And so this is quite striking. And then the other striking feature about this was the long-range stabilization for the local lipid environment. So it wasn't just the annular lipids that were being resolved, the ones that are directly adjacent to the membrane protein, but we're seeing second, third, and even fourth order stabilization for the local lipids. And this was quite striking to us. You can even see this kind of hexagonal packing of the lipid ACL chains, which suggests that the protein was inducing a phase change that was specific to the extracellular leaflet of the lipid bilayer. And when I first started showing this data to membrane biophysicists, they asked an important question, which they should, is what lipid did we use? And for this work, we used a model lipid, DMPC, and we chose this lipid for various reasons. One of them being that these channels had been previously reconstituted into DMPC vesicles and shown to still be functional. But DMPC is an unusual lipid in that it's a completely saturated ACL chain. And so that leads to a relatively low melting temperature of these lipids. And so we became a little bit concerned if this phase change that we're seeing was simply a temperature-induced effect. My argument against that is, if it was simply a temperature effect, we should have seen a phase change in both leaflets. But nevertheless, we wanted to look at this through an orthogonal method. And so again, we turned to computational microscopy. And one of the things you can do with MD is you can take a time average density of your molecular dynamics trajectory. And when we compared the lipid densities by MD and cryo-EM, they overlap almost exactly. And this is with MD run well above the transition temperature of these lipids. And if you looked at the leaflet specificity, it also seemed to match our experimental data quite nicely. And so in blue, I have the order parameter that's consistent with a gel phase of these lipids, and in light blue is the liquid phase. And so this is very striking. However, now we're left with trying to question, what is the potential physiological significance? And coming up with proposals for this is fun. A little bit harder to demonstrate, but I think it becomes even more appreciative if you go back to remember that these channels assemble in these large two-dimensional arrays. And so if you build a back of the envelope model for what this might look like, you can get an appreciation that these channels could effectively be inducing a phase change, or at least stabilizing the extracellular leaflet across a large patch of the membrane bilayer. And so some of the ideas that we're thinking about at this point is that this may reflect some of the asymmetry that's present in biological membranes. The gap junctions are known to be enriched in lipid raft environments. And so perhaps this is involved in lipid sorting to form those raft environments, or vice versa, to partition the gap junctions into these environments. And ultimately, what I've really come to appreciate from this project is that it's really given us a new perspective. And so rather than just asking how are the lipids affecting the structure of these channels, we're starting to ask the question of how do the channels influence the structure and function of the local membrane environment. And we're having fun starting to work with some membrane biophysicists to address this question. And so coming to the end of the talk, I just want to give you a little bit of a glimpse into where this work is heading. I told you about our interest in understanding the functional role of lipids and probing those by cryo-EM. I want to remind us that these channels are dynamically regulated by the cell in response to many different physiological processes. And these dynamic, these gating events are highly dynamic and sample many different conductance and subconductance states. And using cryo-EM, we're starting to resolve some of these dynamic gating features. We don't have the details worked out quite to the level of detail that I'm comfortable going into with our talk, but those details should be coming out hopefully very soon. And I think we're getting a lot of still new insights to gain from these channels using cryo-EM. And so I've used my talk today to illustrate how cryo-EM has had an impact on the field of gap junction biology. But of course, cryo-EM is having a large impact in the entire field of structural biology and many different types of membrane proteins. I like to show this image because I think it really shows where the state of structural biology was heading prior to the cryo-EM revolution. So shown here in gray. And I think what this chart illustrates is that using X-ray crystallography, where we're sort of reaching a ceiling of the new types of structures that were able to be discovered using that technology. And ever since the cryo-EM revolution has started, the number of unique protein structures has continued to grow year by year and has a lot more room to grow. And I really like this sort of mantra that's developed in the field of image all the things. And I really think that the future of structural biology is going to look something like this. And I don't think we're too far away from being able to describe molecular biology at this level of detail. And I look forward to contributing to that and to all of you helping to image all the things. And I know that I'm running a little bit low on time, so I'm just going to move right to my acknowledgements here at the end. And make sure and thank our core facilities, all our collaborators, and all the members of my lab.
Thank you, Steve, for nice and inciting talk. It's great to hear about this gated channel membrane protein. So now the floor is open for the questions. So if anyone wants to ask the question, they can unmute and directly ask the question. So,
hello.
Yeah. Hello.
Hi.
Yeah.
Thanks for the super interesting talk. I had a question about your protein when you put it on grids and whether you had troubles at all with preferential orientation.
Yeah.
Of your proteins
thanks for the question. No, it's a great question, and it's a common pathology with nanodiscs and membrane proteins, at least that we've seen with the centers, is you tend to get this preferred orientation with the nanodisc aligning to the air-water interface. We were fortunate; we really haven't had that issue at all with the gap junctions. I can't explain why. And we haven't noticed a difference. So sometimes those differences come from ice thickness as well, where a little bit thicker ice can sometimes get around that issue. But this particular specimen didn't have that issue at all. So we've been lucky.
That's great.
Yeah. Some common things that people do to overcome that is adding a little bit of detergent to try to break the air-water interface. Fluorinated octamaldehyde tends to be a favorite amongst users in our community to overcome that issue.
Yeah.
Thanks.
Okay. More questions? Zahid, do you want to ask some question? So right now I cannot see any questions typed here.
I might jump in there.
Yeah.
Awesome talk, Steve. Really enjoyed it. You sort of mentioned that it's kind of difficult to trap possible ligands into these things, but with the resolution that you're at, do you think you've got a chance with the right ligand that might have sort of the right kinetics?
Yeah. So thanks for the question. It's a great question. I highlighted how informative this can be for understanding how drugs bind to your protein. But it really points to a limitation or I guess a limitation for gap junctions in that there are really no good inhibitors for these channels. There's common inhibitors that people use in the field. They tend to have KDs in the low micromolar. They're non-specific. They're not specific amongst connexin isoforms, and they bind many other membrane channels. Without showing our hand too much, we've played with these, but the low affinity and low specificity proves to be quite challenging. And so, yeah.
Well, good luck with your future efforts.
Yeah. So low affinity and low specificity leads to heterogeneity, right? And that's always a limitation in structural biology. And the beautiful thing with cryo-EM is its capability to oftentimes be able to resolve some of that heterogeneity, but you can't always do it. So that's kind of where we're struggling with doing small molecule stuff with these channels.
Cool. Thanks a lot.
Yeah, we are running out of time. Before that, we can have a quick question. So, Isabel, do you want to ask?
Beautiful work, Steve. Guess my question was with the other system. I think you quickly mentioned that you use other system, but the nanodisk with nano, and I wonder if you could tell us a little bit more about the system you tried on, why you think they didn't work, or what challenge did you find?
Yeah, so just to make sure I understand, you're asking about the system that we used in the first structural work?
Well, in the second structural work, I think you said you screened quite a few different systems. And but some of them didn't work. And I kind of was wondering what you tried on, why you think they didn't work, what the challenges.
Sure. So I won't say that some of them didn't work, but some of them didn't work as efficiently as I think I think it was MSP 1E1 is the variant that we ended up having the most success with. And so at least with these MSPs, they vary in the types of diameters of the discs that they make, right? And so presumably some of the smaller ones didn't work because they just weren't big enough for the channel. Some of it, once one of them works sort of well, you kind of chase that one. So I can't say that we've exhaustively assayed the effectiveness of all the different MSPs. Once we found one that was working fairly well, we focused then on optimizing the lipid to protein ratios, and that was a bit more cumbersome. What we found to be a really important tool during that optimization process, however, so you usually do your reconstitution and you'll pass it down a gel filtration column, and you'll look to see, the peak of your protein of interest, probably some empty nanodisk peak, and then some void, and you're trying to optimize conditions to increase the intensity of your protein peak. But what can go hidden in those size exclusion traces is the presence of lipid vesicles. And so we also use negative stain-EM to screen the reconstitution of these. And that was really informative to let us know if we had contaminating liposomes in our reconstitution process. So I would say if you're venturing into using these tools, incorporating negative stain-EM as a screening tool is very informative.
Okay, we are running out of time. Thanks for joining the session. There are a couple of more questions, but I would like to thank speaker as well as audience. If somebody wants to leave, they can leave, and I would like to request Steve if he can answer some of the questions and stays for a couple of minutes here. So
Yeah, I'm happy to.
yeah. So there is one more questions from Zahir. Zahir, do you
Yeah,
wanna-
sorry. So did you use like first of all, thank you so much for such an amazing talk, and your presentation was, I mean, it was aesthetically very pleasing. Yeah.
Oh, thank you.
Did you only use potassium ions in your simulations, or like are these channels only selective to potassium? Sorry, if yeah.
Yeah,
or
so
it allow sodium as well or something. Yeah.
yeah, thanks. So they're relatively non-selective towards different monovalent cations. So Bassam is followed this work up, and we've done in silico electrophysiology with many different ions, and we're writing that work up now. But what we effectively find is, and it's consistent with experiment, that they're relatively non-specific between different types of cations, but they select fairly well between cations and anions.
So there is no selectivity filter in these channels, right? Yeah.
Not to the point of discriminating monovalent cations. So the pore is just too big, I think, to be able to do that. The ions come in completely solvated, and so the differences in conductance that we see are consistent with differences in their diffusivity.
You mentioned monovalent. So do you think that the divalent ions have trouble crossing these channels?
Yeah, so experimentally it's been shown that these channels can permeate, for example, calcium ions. We haven't simulated calcium. Calcium has a complex role of gap junctions. It can permeate at lower concentrations, but at higher concentrations it inhibits the channels. And so that's an aspect that we're interested in as well.
Thank you.
Yeah, of course.
Yeah. Thank you. Thanks a lot, Dr. Steve, and thanks all of the audience who have joined today's session. And if they have more questions, they can reach Dr. Steve by email. And I would like to remind that our next session will be on 14th of November. So yeah, that's a great talk. Thank you. Thanks, Dr. Steve.
Oh, thanks so much. It's my pleasure.
Yeah.
Thank you all.
Thanks you all. Thanks.